This method can help answer key questions in the stem cell field about the role of adult stem cells in the regulation of cancer cell behavior. The main advantage of this technique is that it allows the evaluation of cancer cell and stem cell interactions in vitro without the use of living organisms. This technique has potential for cancer metastases as it models the promoting role of adult stem cells in cancer metastases to hard tissues, such as bone.
Demonstration of the procedure will be carried out by Hüseyin Apdik, a postdoc from my laboratory.
Begin by using sterile extraction forceps to remove the pulp tissue from the center of wisdom teeth obtained from young adults aged 17 to 20 years old. Place the tissue in cold complete DMEM in 10 centimeter tissue culture dishes and use a scalpel to mince the samples into 2 to 3 millimeter fragments. Transfer the pieces from each dish into individual wells of a tissue culture treated 6-well plate and cover the tissues in each well with 200 microliters of complete DMEM. Allow the tissues to attach to the well bottoms with an at least 2-hour incubation in a humidified 37 degrees Celsius and 5% CO2 cell culture incubator. At the end of the incubation, feed the tissues with 2 to 2.5 milliliters of fresh complete medium and culture the cells for an additional 8 to 9 days. When the cultures reach 80% confluency, wash each well with 2 milliliters of PBS, followed by detachment of the cells with 2 milliliters of trypsin at 37 degrees Celsius. After 2 minutes, stop the reaction with 2 milliliters of complete DMEM per well, and collect the cells by centrifugation.